If you would like free handwritten notes on this topic, please head over to my site. You can find the link in the description below. In this video, I'll be continuing on with the immune system topic. If you have not seen my previous videos on the introduction to the immune system, or humoral versus cell-mediated immunity, please give them a watch first to better understand this video. I'll link these here now. In this video, I'll be going through immune responses. So, what are immune responses? We have primary and secondary. With primary immune response, this is when the pathogen enters the body for the first time, and it is about how the body responds to this. As I mentioned in previous videos about the antigens on the surface of pathogens being detected as foreign, when the pathogen enters the body, the immune system detects the pathogen as a foreign intruder. Now, in order to get rid of that pathogen, the immune system needs to have the B cells with complementary receptors to those antigens on the pathogen. However, we only have a small number of B cells that contain those receptors, and it can be hard to find those B cells. It is like having a pile of keys and trying to find that one key that can open the door. So, finding that B cell is the most time-consuming part of this process, and this is called clonal selection. Once those B cells with the complementary receptors are found, they rapidly replicate via mitosis. This process is known as clonal expansion. Then, some of these cells will turn into plasma cells, which will create antibodies to deal with the pathogen, and others will turn into memory cells to remember the pathogen if it tries to infect you again in the future. So, once the levels of antibodies in your body rise, the pathogen is eliminated and you feel better again. Now, secondary immune response, this is where the pathogen re-enters the body. Again, the immune system detects that the antigens are foreign, but the B memory cells also remember that they have seen these antigens before. So then clonal expansion is triggered a lot sooner, which means that plasma cells are made sooner and the antibodies are produced quickly to deal with the pathogen. So the host doesn't even realize that they were infected because the pathogen was killed before the host could even get ill. If you have a look at this graph, you can see the primary and secondary immune response. Primary immune response will take some time before a spike in the antibodies is going to be seen, compared to the secondary immune response. But also the amount of antibodies produced in the secondary immune response is much greater too. Now, if I give you an example, have you ever had chickenpox? If you have, when you first had it, that would have been your primary immune response to it. And you got really ill and you felt really itchy because you had all those red spots all over your body, because your immune system took time during clonal selection and expansion before you could feel better. But now you have memory cells, so when you are older and if your child gets chickenpox, you will not catch it again because your memory cells will be triggered to prevent you from getting ill. But on the flip side, if you think about how many times you get a cold, the reason for that is because yes, although when you get your first cold, the same steps occur. The issue when you catch another cold in a couple of months or a year, this is because the influenza virus that causes colds are constantly changing their antigens on their surface. So when they re-enter your body, your immune system detects it as a different pathogen because they have changed their antigens. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.